Je pense que je vais faire une moitié français, moitié anglais. Comme ça, tout le monde va en avoir un peu. So, thanks for staying awake. And it's been a long day, but this is so exciting. This is all about the drinking water stuff that actually nobody cares about. That's what I learned this morning. So it makes me a little sad. Well, I do care about it, and most people do, so. C'est que je vais vous présenter l'avancement des travaux pour les étapes 2.1 et 2.2 qui visent à identifier les points critiques d'accumulation de cyanobactéries dans les usines de filtration. Basically, cyanobacteria enter the plant and they accumulate in different processes. And although there is risk with what comes in, actually the accumulation in the water treatment plant is the key thing to remember. That is where numbers of cyanobacteria can grow by 100-fold, 1,000-fold, and if they contain toxins, obviously intracellular and possibly extracellular toxins would be released into the treatment plant, which is not good news. Le deuxième, 2.2, c'est le traitement des cyanobactéries dans l'eau et les boues. So similar to what Sarah was talking about, so basically it's a little less messy than in the source, but not so much because in a water treatment plant there are many things going on as well. Operators need to produce drinking water. They're not doing a research project, so things do change. And our focus is to see how we can help them and actually see full scale and in controlled fashion at the lab, same as Sarah, what happens when we do apply treatment, mostly oxidative treatment. So, a big team for Arash, myself, and Sarah and Benoit Barbeau being involved. Liama working on probes. I'll be presenting most of that work. Farad working on sludge, so focusing where the cyanobacteria do accumulate. Saber working on oxidation, and you saw his face twice, so he's also working on oxidation in the source. Then we have our tech and lab support, Research Associate Jacinthe Mailly, Détiana Eliard, and Yves Fontaine, which cover all our many needs in the field and in the lab. Choukoufé, Julie, also in the lab or helping out here, and Anna also showing up in every slide because she's our translator between the team of Jesse and this engineering team. So the research aim is for the first part of it, is to apply the online cyanobacteria monitoring tools for real-time treatment adjustment. So the idea is to understand and be able to predict when things could go bad in treatment plants. Not only knowing what comes in, but understanding how much may have accumulated, so you need to react proactively and address this reservoir of accumulated cells and toxins. So we said we needed to identify the gaps in understanding cyanobacterial-related issues for water treatment, and we did that through a combination of lab experiments, which I will not be showing, but Arash will be showing a little bit of that, and full-scale applications. So I'll be focusing on the full-scale and the tool. All these very exciting tools, advanced toxins, genomics, that's all, and no, it's all great, but we also need in plant operation an online tool. We don't have an advanced online tool, so we need these probes to understand how things fluctuate hourly, daily in the plant, and relate those measurements and the measurements of simple things like oxygen, like turbidity, and how do those relate to what we're seeing in terms of advanced results. So one of the first things we did is to look at across the world, including in Quebec, there are guidance when a certain number of cells is reached. You have to react as a plant operator, so there are called alert levels. So depending if it's alert level 1 or alert level 2, you will have to react differently. These are based on cell counts. We know cell counts are not good. We've done a lot of work here when Arash was here a number of years ago, and Sarah's done work to show that biomass is a better indicator, but basically we want to say, when do things get to a level that there may be a potential for enough toxins to be produced and released to give us problems? So what we have proposed over the years is to use the zones, the probes to do that, but we need to understand how these probes work. And actually, they give us really nice measurements. You could have a measurement every half hour. We tend to cycle them at different points in the plant, so it's every 2 hours. But whatever is the case, you need to understand is one number, as you see here, for example. So you would have, let's see here, one big number here. What would that mean? Like that would be, you would have this here, and this would be the uncertainty between the two red boundaries here. And basically you need to understand how precise is your signal, but also what does a peak that lasts for an hour actually mean in terms of risk? Is it direct breakthrough? No, we have process in the plant, so it's not direct breakthrough. So we need to understand what is a real peak, what is a meaningful peak in terms of intensity and duration, what is the error of the measurement? And I'll show you a couple of the last errors we measured. Arash has done a career of measuring errors on different probes. And we had a really good rock on this project. They asked a couple more questions. We addressed them, and I'll show you results. So what are the last ones that we thought needed a visit again? So natural organic matter, when they asked me to do that, I said basically they don't interlap. So the spectra don't interlap, shouldn't be a big deal. But we did go through and we found that, yes, it can have a small incidence. So we want to understand how to do that. We're going to measure pigment. We're going to take a natural organic matter, measure the probe's response, put cells in there and add the gnome, natural organic matter, and see if anything happens. Does the gnome coat the cells? Does it interact with whatever happens in that mixture? We also looked at water temperatures. We've experienced blooms in Canada at lower temperatures. Does it change between 30 and 10? That's a pretty valid question, and basically just about any probe has a temperature effect in it, and it should have a correction if that is intense enough to end cell aggregation. So this is the first set of results you're seeing here. On the y-axis, you see the ficocyanin in our RFU reading, so that's a raw reading from your probe. And you can see here 3 concentrations here of gnome, so natural organic matter. Here was Suwannee River natural organic extract. That's the classic one, well-defined spectra for emittance and absorption, so a good candidate. And what you see here are different measurements with, you can see here with different concentration, Microcystis aeruginosa, different color, no gnome, gnome at 0.2, 2, and 10 milligrams per liter. So 10 milligrams per liter is not off the chart, but it's a substantial amount of gnome. So you can see that without any cells, the numbers are very low. So we're talking a very, very small effect. With cells, very little. And we tend to see something happening when you get to the higher gnome concentrations. The signals go down. We don't know why exactly, but this could be important because you're talking here of a difference of signal of, let's say, a couple of RFUs, and some of the other cells we tested up to 5. That could be enough to push you up the boundary of an immediate type alert level. So this is important to know. So this is temperature. So here again, so Microcystis aeruginosa, just a suspension. And then the different colors, 10 degrees C in blue, 20 degrees C in orange, and 30 in gray. So you're not seeing much of a difference, except when you have high cell numbers. So we're going to repeat this experiment, try to understand. That is important because you really want these probes to work well. When the cell numbers are high, that's when you're really keen on knowing how much they are. So we're going to repeat that. We've done it with different species, and we're going to try to understand. Here we're talking a difference of about 3 RFUs. Not the end of the world, but again, you're at the point where you want to know. This is just one of the errors of these probes, so you want to understand and take this into account. So I think the largest progress we've made since our last meeting, Arash had nicely presented the new system, which I'll show you in a minute, was to deploy these probes to measure at different points of the treatment plant. So you coming in the plant, after coagulation and settling, etc., to see how well the cells are removed. So this table here summarizes the plants for 2017, then below 2018, all year or just part of the year, in which we deployed these probes to measure the RFUs at an interval of anywhere between 10 and 30 minutes for between each point. So this is the famous device. When we started, we had a homemade device that wasn't working perfectly. I think now we have an extremely nice device, well automated, online measurements. You can see what's happening from a distance, which is nice. And the 3 prototypes were purchased in 2018 and deployed in 3 plants. So we have data loggers, telemetry, set with a modem system, and it really provides, you can see a screenshot of what type of access you can get to online data. So what does that bring you? So if you're looking at a time series of these probe measurements, which some of you may have seen, the purpose of what we're doing is to relate this to these famous thresholds you want to see. So you want to develop how you will calculate when you reach a threshold, and those are the reference thresholds that you would action thresholds. And in the little dots here would be the counts that we had run parallel to these online measurements. So for this, what we can say right now, the probe systems measuring at several points in alternative ways. On séquence le prélèvement des échantillons de l'eau brute jusqu'à l'eau traitée work very, very well. We have a number of events for which we have detailed cell counts, and also we have collected data for samples for genomics that will accompany this. What this says, in the different colors here, in green you can see that's the raw water, that's the highest peak. The second one would be the clarified water. And basically what LIA will be doing is not only treating the signal, matching these signals in time, but also deciding how long a signal is a significant enough breakthrough throughout the plant to mean anything in terms of risk. And risk being the possible presence of toxins in the sufficient condition to create any kind of real challenge to the plant. So the probe signal interventions, for interferences, we found that GNOME had a small impact, a couple of RFUs, but not negligible. So yeah, that was a valid comment from the ROC. Actually, when we put cells, it's a lot more. So there's this interaction between the cells probably and the GNOME that we don't have to figure out. Temperature had a significant decrease in signal from 10 to 30. So there's something to think about. If that's important, we need to understand it. I've worked with probes for the last 10 years quite a bit. We just get the manufacturer to take that in and correct the signal with this information. It's not difficult. For the treatment of online data, we're looking at statistical analysis using time series analysis for the 2007-2018 data. We're just getting into that right now. Elia has finished her lab work just about. And we're going to determine how probe signals can be used to predict conventional treatment breakthrough. So can they be relied upon to see that there's significant breakthrough? We have to define what's a significant breakthrough. That's a big question. And secondly, can we use, I think that's more important, can we use these probes to evaluate how much we accumulate in sludge and whether we need to act on sludge? And finally, we're going to just dive into the multi-parameter data. There's a lot of genomic data. I'm always impressed with the millions and numbers of billions or whatever. We have hundreds of thousands of points of data that we need to correlate with speciation, with genomics. And that's, I think that's you starting here.